Welcome everybody. Today we'll be looking at the classification of bacteria based on key traits like shape, their staining, methodology, metabolism, and genetics. This scheme of bacterial classification helps scientists to identify bacteria and to study them and to also understand their bacterial behavior, especially in microbiology, the medicine, and ecology. So let's go briefly into this topic and see how the microbial world is identified. In understanding of bacterial classification, it is a fundamental aspect of microbiology and it plays a crucial role in how we study and identify and understand this microorganism. Bacteria can be classified into different groups based on several keys, and these include phenotypic similarities. When we say phenotypic similarity, we're talking about the observable traits such as shapes, staining characteristics, and colony morphology. Then we also have physiological or biochemical relatedness, which involve the uh, metabolic activity of the bacteria with respect to the nutrients, how they metabolize the nutrients, the enzymes they produce, or how they respond to environmental conditions. The evolutionary relationship are determined through genetic analysis, and it helps to trace the lineage and ancestry of bacterial species. When we talk about lineage and ancestry species, we talk about dating back in their lineage, how they evolve and how they were being, evolving from one generation to another. So each of these classification methods offer unique insights and serve as different scientific and practical purposes. For example, the phenotypic classification is often used in clinical settings for quick identification of bacteria, while evolutionary classification helps researchers to understand bacterial diversity and origin. You see, because bacteria are incredibly diverse and adaptable, scientists have developed multiple typing schemes to group them effectively. And these schemes are not mutually exclusive. Rather, they complement each other depending on the context, whether it is medical diagnosis, whether environmental studies, or evolutionary research. Now, in scheme of classification of bacteria, there are key criteria for bacterial classification. That is, when classifying bacteria, there are some keys, some observable keys of, let me say, some key points, key information that scientists often rely on. And this includes observable or testable traits. These traits can help distinguish one bacteria species from another, and it can provide clues about their behavior, their habitat, their potential impact on human health, and so on and so forth. So let's look at the first key, which is the morphologic characteristics. When we talk about morphologic characteristics of bacteria, we are talking about the physical appearances of bacteria under the microscope. And these are how we see them, how we observe them under the microscope. We have a shape of bacteria which can be cocci, spherical shape, that you can see on the left hand side of this slide. The cocci bacteria are seen in this shape. We have the Coccobacillus. We also have the Vibrio, then we have Bacillus, we have Spirochete, and we have Spirillum. These are shapes of bacteria under the microscope, and they help us to differentiate them from one another. Then bacteria can also be arranged, whether they are Coccobacillus, whether they are cocci, whether they are Bacillus, they could be arranged. The arrangement also determines the type of bacteria. For instance, some bacteria can be arranged in chains such as Streptococci. Some that are spherical in nature, they can be grouped together in clusters. They appear like grapes. Those are Staphylococci. and some can appear in pairs. Cocci in pairs are usually called diplococci. Then also we have the staining characteristics of bacteria, which is a major tool in classifying gram-positive bacteria and gram-negative bacteria. This is a characteristic differentiation of their cell wall structure, whereby gram-positive has much peptidoglycan, while the gram-negative have lesser of this structure. It is the basis for their differentiation. Then we also have the presence of structures such as flagella for movement, pili for attachment, or capsule for protection in their morphology. That is for morphology. Then we look at the growth characteristics, which involve how bacteria grow in culture and under different conditions. When bacteria grow, they form colony, and the colony can take different shape, color, or texture. The size of bacterial colony on agar also depends on the nature of the bacteria. Some can be very small, some can be big, and so on and so forth. So this colony formation for their growth characteristics is a way of identifying them preliminarily. We also have the growth rate of bacteria. Some can multiply rapidly while others grow slowly. They also require oxygen in terms of growth. Those that are aerobic need oxygen, while those that are anaerobic, they can grow without oxygen, including facultative and microaerophilic. We'll still get to that one later on. Then their temperature preference and so on and so forth. We'll get to that later. Then we have the antigenic properties. Antigenic properties are surface molecules that bacteria possess, and it can trigger immune responses. These molecules are known as antigen because they can trigger immune response, and they vary between species of bacteria and strains of bacteria. We have the capsular antigen, which is K antigens. We have the flagellar antigen, which is H antigen. We have the somatic antigen, which is O antigens. These properties of bacterial antigenic properties are very crucial in serotyping, which is used in identifying specific bacterial strains that are usually involved in some epidemiologic or involved in some kind of disease. Some of them are actually also used in vaccine development. The next key identifier is the biochemical characteristics. These refer to the metabolic activities of bacteria, such as enzyme production, e.g., they can produce catalase, they can produce oxidase, urease, and coagulase. These enzyme production are specific to groups of microorganisms and bacteria. So we use them also in identifying them. They also carry on some fermentation properties, which is also a form of biochemical identification. Some are able to ferment glucose, lactose, or other carbohydrates. Some can produce gas during their growth, and these are used in identifying them from one another. Then we have the pathogenicity and toxin production. Some bacteria are actually harmless, while others are harmful. Some cause disease, while some are used in preventing diseases. So they have different uses. The ability to cause disease or to cause infection is usually linked to them having what we call virulent factors. These virulent factors include certain substances such as adenosine, invasins, and some enzymes that can actually damage the host tissue. It allows them to penetrate the host tissue. It allows them to get deep into the host and cause damage. Some bacteria also produce toxins. These toxins are secreted by different types of bacteria. We have the exotoxin and we have the endotoxins. And these are components of their cell wall. The presence of toxin can act in a cell of healthy human being can lead to severe symptoms. So when we understand these traits, it's essential in clinical settings to determine the severity of infection and to guide treatment strategies when microorganisms are involved. So, morphology and staining reaction of bacteria. When we talk about morphology, I've explained briefly about morphology the other time because it's one of the principal methods that you use in classifying bacteria, and it's by examining their morphologic characteristics. That is, their physical structure and their appearances under the microscope. In bacterial morphology, some of the important thing is that the size of each bacteria is very important. Bacteria vary in their dimension. A typical bacteria range from 0.2 to 2.0 micrometers in diameter, while a smaller bacteria can be very smaller in size than that. So in terms of their arrangement, you can see how bacteria are arranged. This is a single coccus. These are cocci, streptococci, you can see sarcina, you can see staphylococci. They are arranged. In this arrangement, help us to identify them. Chain bacteria in cocci, example, we have streptococcus. Those that appear in cluster, we have staphylococcus, and those that come in pairs, we have diplococcus. For the growth characteristics of bacteria, beyond their microscopic appearance, it is very important for us to classify them based on how they grow in culture. So for bacterial scheme of classification, scheme of bacterial classification, we also look at their growth in culture. When we culture them in microbiology, culture is a way of growing microorganisms. Unlike when we say culture is people's tradition, in microbiology, our culture is the way we grow microorganisms. And their growth characteristics provide insight into the organism physiology and environmental preferences. And this play a crucial role in their identification. Now, in terms of their colony appearance on media, bacteria can provide different circular, irregular, or filamentous shape on culture. We have those that have a pinpoint to large size. You can see a bacterial size that is like a pinpoint, whereas some can be large and circular. In terms of color, they can come in different color. We call it pigment. We have white, yellow, red pigment. And this is also a form of identifying them because they will be consistent in production of their pigment. A white pigmented bacterial colony will always provide a white pigment, yellow, red, and so on and so forth. In terms of their texture of the colony, how they grow, some can provide a smooth colony, it looks smooth, while some can provide a rough colony. Then some can look mucoid. And in terms of their elevation, some can be flat, some can be raised, and some can be lobed. So these are distinctive features that help to differentiate bacterial species. Then also in their environment, in the medium where they grow, bacteria can also alter this medium based on the breaking down of the constituent of the medium. For instance, they can cause hemolysis. Hemolysis is the breaking down of red blood cells by bacteria. So they can form different types of hemolysis. We have alpha hemolysis, we have beta or gamma hemolysis. This type of hemolysis is typical in classifying bacteria. There are some bacteria, when they grow on blood agar, they provide alpha hemolysis. If you can see, alpha hemolysis has a greenish colony around it, while beta hemolysis, this you can see here, and gamma hemolysis is not really conspicuous. So this is a type of three type of hemolysis that that bacteria exhibit on blood agar, and it is a form of classifying them also. I've said something about pigment production before. They grow with pigment production. This is like color. So a very distinct organism that grow with also color is Pseudomonas aeruginosa. It gives a greenish coloration on agar. Then we also have pH changes, which is indicated by the color of a pH sensitive media. There are some media that are pH sensitive. So when a bacteria provides a change in pH of a particular media, such media has been designed to be pH sensitive. So the media will indicate this by change in color. So these are also a way of identifying bacteria by their growth in the surrounding medium. Let's go to the next aspect, which is their growth in liquid medium. Liquid medium is also called broth. This is an example of a broth. It's a liquid medium that facilitates the growth of bacteria. In broth cultures, bacteria can exhibit different growth patterns. They can have a uniform turbidity, which becomes cloudy. A turbidity is seen by most everyone when you see the bacterial growth in let me say some food item like liquid food that's supposed to have a clear, transparent appearance. Whenever it is turbid, you know it is contaminated. So bacteria can actually give you a uniform turbidity in growth, and it can be cloudy. You see the broth become so cloudy. Then they can also give you a pellicle formation, pellicle. That is, they provide a film on the surface. They provide a film on the surface of the broth. That is the way they grow, and you call it a pellicle. We also have flocculent. Flocculent growth is the clump. The clump is suspended in the liquid. You can see their growth in clumps, and it's suspended around in between the broth medium. Then they can also form a sediment growth. Sediment. This sediment, they grow and they form sediment under the broth medium. So this pattern reflects different type of how bacteria interact with their environment and cause growth. So their growth in liquid cultures can be either uniform growth, pellicle formation, sediment, or flocculent growth. Don't forget that. So in terms of oxygen requirement, bacteria also grow. I told you in the beginning that we'll still talk about this. We have aerobic bacteria, aerobic organism that grows with requirement of oxygen. This is a primary distinguishing characteristic of bacteria, how they respond to oxygen. So aerobic bacteria require oxygen to grow. Anaerobic grow in the absence of oxygen. Then we have the facultative anaerobes that can grow with or without oxygen. Facultative anaerobes can grow with or without oxygen. And we have microaerophilic that require little oxygen. They require little concentration of oxygen. Excuse me. Example of microaerophilic is Campylobacter jejuni, Helicobacter pylori. Then we have the obligate aerobes that require oxygen for growth. They require oxygen. Example is Pseudomonas aeruginosa, Mycobacterium tuberculosis. We also have obligate anaerobes. Obligate anaerobe cannot tolerate oxygen and they die in its presence. Example, we have the Clostridium tetani, which is obligate anaerobe, and Bacteroides fragilis. So we have the aerotolerant or facultative anaerobe, which can grow, ferment sugar in the absence of oxygen, such as E. coli. It can grow with or without that. Then their growth characteristics is important in assessment of bacteria in terms of how they incubate them, the temperature they require, which is also used in determining the nature of the bacteria. So some bacteria can grow well at a higher temperature. Example is Campylobacter jejuni, which grow well at 42 degrees centigrade in the presence of certain antibiotics. We also have Escherichia coli and most other Enterobacteriaceae which can grow on minimal medium. Minimal medium means the medium contains some not too enriched substances. It is not fortified with adequate nutrients, so they can grow on it, while some other organisms will not grow because they require adequate, sufficient enriched medium for them to grow. So, bacteria can also be identified based on the temperature requirement. We have the temperature requirement for bacteria under diverse environmental conditions. Their growth can be classified based on temperature, salinity, pH, antigenic properties. But for temperature, we have thermophiles. Thermophiles are organisms that are actually found in hot springs. They grow best from 45 degrees to 80 degrees. They are found in extreme heat environment like hot spring, like I said, and we have Thermus aquaticus, which is known for its heat-resistant enzyme used in PCR. We use its enzyme in molecular biology. We also have the mesophile. Mesophiles are actually the most common bacteria, and most human pathogens are classified under this group of bacteria called mesophile because they grow or they prefer to grow at moderate temperature, which is between 20 degrees to 45 degrees, or let me say 20 degrees to around 42 degrees are mesophiles. At this temperature, a large number of microorganisms are found. Example, we have E. coli, we have Staphylococcus, and so on and so forth. Then we have the saprophytes, which thrive in cold environment. That is, they like to grow in cold environment below 15 degrees centigrade. They are found in deep ocean water and polar region. So example of saprophyte is as stated below. They are adapted to survive in permafrost. In terms of salinity, microorganisms can also be classified based on their salt concentration adaptation requirement or tolerance. We have in this basis of their salt tolerance, we have what is called halophiles. Halophiles are those type of bacteria that require high salt concentration for them to grow. They are found in hypersaline environments like the Dead Sea. Everybody knows the Dead Sea is rich in high salt content where no organism can survive, but halophiles are found surviving in this environment. We have those that are called halotolerant, means they can tolerate a wide range of salt concentration. Example is Vibrio cholerae. Vibrio cholerae tolerating wide range of salt concentration is different from requirement of high salt concentration. Tolerating salt concentration means at different ranges of salt concentration, whether low, medium, or slightly above medium, they can still tolerate it. Okay, now Vibrio cholerae can also live in brackish water, and they can also live in human host, because the brackish water salt content is different from human host. That is why they are halotolerant. We also have those that are classified based on their pH level. That is, we have the acidophiles that thrive in acidic environments, and we have the alkalophiles that prefer alkaline environment. That is, they like to live in pH above 9. Example, we have the Bacillus subtilis that is used in biotechnology and cleaning products. Bacillus subtilis, that is the name of the alkalophiles. And the acidophiles, we have Bacillus thuoxidans that are found in acid mine drainage. I mentioned briefly the other time about the antigenic properties of bacteria. Yeah, bacteria can also be classified based on their antigenic properties. It is a kind of specificity of antigen and antibody reaction that has been used in bacteriology as a means of identifying different strains of bacteria. And they have this structure that are called antigen, which help them in triggering immune responses. They have the capsules, they have the flagella, they have the cell wall, and other anatomical elements that are found in their cells. When these components are introduced into animals or humans, they stimulate the production of specific antibodies. So the resulting antisera can then be used to identify the bacterial strain. Let me come again. When these organisms or their components are introduced into animal cells, animal blood, they will stimulate the production of specific antibodies in response to their presence. So these specific antibodies, which is specific to the components or this particular bacteria, can then be used to identify the particular bacterial strain. So to classify bacteria based on antigenic composition, it helps to detect pathogenic strains. It also aids in vaccine development. For instance, Salmonella is classified into serotypes based on its cell wall O and flagella H antigens, which is used in identifying it in the hospital or laboratory. Streptococcus pneumoniae has over 90 capsular types, each with distinct antigenic profiles, and these are used in determining their presence whenever they cause an infection or whenever they are present in a specific type of epidemiological outbreak. So the serotyping is also used to distinguish strains of exceptional violence or organisms of public health importance. For instance, Vibrio cholerae 001 is the pandemic strains, and E. coli, enterotoxigenic and enteroinvasive or enterohemorrhagic and enteropathogenic serotype are usually known for causing different types of infection, and they can be identified based on the serotyping. This serotyping helps us to differentiate individual E. coli or differentiate Vibrio cholerae that is of pandemic strain. So whenever individual or there's a problem or there's an outbreak and there's a call for need for investigation, these are things that are investigated, the antigenic properties or serotyping of the organism, to know if the strain involved is an epidemic strain. That is, if it's a strain that can cause a wide range of infection in a particular locality. We'll be rounding up soon now. Biochemical characteristics, I've talked about that previously. That is, microorganisms are classified based on the types of enzyme they produce and the way they carry out fermentation and so on and so forth. So classification based on biochemical tests. For other organisms, we have the gram staining, which is what I've explained previously. It helps us to differentiate gram positive and gram negative bacteria. This is based on biochemical tests and their mode of nutrition, which is also one important aspect. We have the phototrophs, which require energy from the light. They require their source of energy from the light, because in classification based on nutrition, breaking down of nutrients and the use of energy is very important. So phototrophs use energy from the sunlight. The photo. You say anything when you hear photo, you know it has to do with light. And they use various carbon sources. And among the types of photolithotrophs, we have those that use light energy and inorganic compound as their carbon source. We also have the photoorganotrophs, which use light for energy and organic compound for their carbon sources. The example is there, the Chromatium ocani. You also have the chemotrophs, which use, rather than light, they use chemical compound as their own energy source, and their carbon sources also varies. Among these, we also have the chemolithotrophs and the chemoorganotrophs. The chemolithotrophs use inorganic chemicals such as ammonia, hydrogen sulfide for energy, while the chemoorganotrophs use organic chemicals for energy. Example, we have the nitrosomonas that oxidize ammonia for energy. Then we have the autotrophs, which use carbon source. They use carbon dioxide for their own energy source. For their carbon source, but their energy source varies. Autotrophs are those that actually use carbon dioxide as a sole source of carbon to prepare their own food. Photoautotrophs, among the types of autotrophs, use light energy to fix carbon dioxide. Example is the cyanobacteria. And we also have the chemoautotroph that use chemical energy to fix carbon dioxide. Example is Nitrobacter. We have talked about this previously in our other classes. Then we have the heterotrophs. The heterotrophs are those bacteria that use organic compound as carbon source. They lack the ability to fix carbon dioxide, and most human pathogens are usually found in this category. Most of them are heterotrophs. They don't fix carbon dioxide, so they are heterotrophic in nature. Some of them are simple because they have simple nutritional requirements. However, we have some of them that require special nutrients, and we call them fastidious organisms. So fastidious heterotrophs are those that require additional enriched medium for their growth. So long, these are the classification scheme for bacteria, and I hope you have been able to enjoy the lecture so far. We appreciate if you subscribe, like, and share. Thank you.